So you've just heard from Madam Kraut describing a number of the emergent technologies for next generation or second and third generation sequencing. So what I will be doing now is going into some detail on a specific application of these technologies, specifically the analysis of antibody repertoires for the discovery of biologics and for the pursuit of discovery of underlying disease etiologies. My talk will basically, am I being too loud? Can you hear me? Well, okay. What I'll do is provide a brief introduction into where I work and why we are maintaining an active interest in antibody repertoire analysis. I'll go through a brief introduction into the molecule itself to motivate why we're interested in it. And then I'll go through some of the pragmatic considerations for appropriate analysis of antibodies, analytical methods, and then applications of next-generation sequencing into artificial phage display library repertoires and natural repertoires. The objective of this talk is to provide a pragmatic approach, so please ask me if you have any questions, and I'm going to try to provide our specific examples of the algorithms, reagents, and analysis approaches that can be applied to this class of questions. So first, I'll just describe a little bit of where I'm working. So Pfizer has the Renat laboratories in South San Francisco. We do biologic-based development and have large biology groups. So we're interested in antibodies both from the perspective of them being our drug, but also from the perspective of analyzing the adaptive repertoire in oncology, autoimmunity, and other disease states. We have a number of groups. I'm coming from the protein engineering department, where we have a variety of mechanisms for extracting antibodies to develop. We also work closely with the Applied Quantitative Genotherapeutics group, which works with human genetics for patient stratification and biological discovery. So this is going to be a brief introduction into the molecule that I'll be discussing for the rest of the talk. This is an antibody. It's essentially nature's solution to molecular surface recognition. Your body produces a large number of them. You have somewhere on upwards of 1 billion novel surfaces at any given time. And that ability to recognize novel surfaces makes them an attractive scaffold for drug development. So the new class of biologic drugs have become quite popular in the market because of their low side effect rate and high precision on targeting their target surface. So you can divide an antibody into essentially two areas, the separate functions. You have the constant domain, which is this area down below. What this is doing is your standard molecular surface recognition. This basically provides a tag where other areas of the immune system can come and respond to whatever the antibody recognizes. It's this area up at the top, so the FV, or the variable fragment, that does the actual recognition, that recognizes novel surfaces. And that's what we'll be talking about today, is what are the mechanisms that nature has employed to be able to allow this to detect a surface it's never seen before, and how we can query into that diversity using high-throughput sequencing technologies.